In this video, we are going to learn about one of the tools of recombinant DNA technology, and that is enzymes. There are different types of enzymes concerned with the genetic engineering. To conduct any genetic experiments, we need DNA, and DNA is present in the nucleus of a cell. If we want to obtain this DNA, we have to open the cell. For opening the cell, there are enzymes and they are called lytic enzymes. So the first one is lytic enzyme. So if a bacterial cell is there and we want to obtain bacterial genome, then the enzyme used is lysozyme. It is lysozyme. If a cell is plant cell, then the enzyme used is cellulase. And if it is a fungal cell, the enzyme is chitinase. By using these enzymes, we open the cell and the DNA is obtained. Suppose the obtained DNA is this. This is the DNA which we have obtained by using lytic enzyme. And this DNA, particularly at this part, it contains certain genes which we want to introduce into the host cell to obtain products which are useful to us. To obtain this fragment of the DNA, we have to cut the DNA. For cutting this part of the DNA, fragment of DNA, there are other enzymes, and these enzymes are called cleaving enzymes. Cleaving enzymes, they cut the DNA molecules into fragments. And there are three types of cleaving enzymes. The number one is exonuclease, second is endonuclease, third one is restriction endonuclease. These are the enzymes used for cleaving of DNA molecules. So here we will see one by one. The first one is exonuclease. Exonuclease, it removes the terminal nucleotides from the terminal end of one of the strand of DNA duplex. It removes the nucleotides from the nucleic acid. So it removes only from the DNA. It doesn't cleave RNA. RNA is not cleaved by exonuclease. The second type of the cleaving enzyme, that is endonuclease. Endonuclease, it breaks the DNA molecule from any site except the terminal site. So these are the terminal site. So except terminal site, it removes or it breaks the DNA molecule at any site, and apart from this, it cuts the either single strand or double strand. This is the characteristic of endonuclease, and RNA is cleaved by endonuclease. Restriction endonucleases are also referred as molecular seizures, and restriction endonucleases, they cut the DNA sequence in such a manner that they come to possess short single-stranded free end, and that end is called sticky end. For example, this is the enzyme, restriction enzyme EcoR1. It is obtained from E. coli, which is present in the colon of human being. Restriction enzyme, it acts on the specific palindromic sequence, and that is GAATTC, and the complementary, that is strand. Here also it reads the same. If you read both the sequence from this direction, 5' to 3' direction, and here also from 5' to 3' direction, you can read these two sequence of nucleotide same, and that is called palindromic DNA sequence. So eco R1, it acts between G and A. Here also and here also you can see in the complementary opposite that DNA strand, it acts between G and A. Restriction enzyme is the characteristic of prokaryotes. It is characteristic of prokaryotes, and for prokaryotes, restriction enzyme, that is a kind of natural defense mechanism against the bacteriophage. Restriction enzyme was first isolated by W. Arbour. He was working on host control restriction of bacteriophage, and he observed that some bacteria, they were able to protect themselves against the bacteriophage, and he concluded that this occurs due to two enzymes, that is restriction enzyme and the modification enzyme. Restriction enzyme, it acts on the specific sequence of phage genome and cut it into pieces. This is the phage genome, and the restriction enzyme of the bacteria, it cleaves the phage genome into pieces, into small pieces, and hence it restricts the propagation of phage into the host bacterium, and that's why it is called restriction enzyme. And the second is the modification enzyme. Modification enzyme is methylase. So restriction enzyme, which is present in the bacterium, they also recognize the same sequence present in the genome of the host cell. This is the nucleotide sequence present in the host genome, and restriction enzyme is recognizing the same sequence. So it can happen that the restriction enzyme can cleave the host genome. Just to prevent cleavage by the restriction enzyme, certain nucleotides, they get bonded with the methyl group, and so the restriction enzyme, it fails to recognize the same sequence. Arbour hypothesis was later verified by Hamilton Smith, and it was applied in the genetics by Nathan. Hint 2 was the first restriction enzyme isolated. Restriction enzymes are of three types. Amongst them, the second type of the restriction enzyme is widely used. It can be used in vitro, and it doesn't need ATP for the cleavage of double strand of DNA molecule with specific sequence, and this specific sequence contains 4 to 8 nucleotide. It's important. It contains 4 to 8 nucleotides, and this sequence of 4 to 8 nucleotides, they are known as restriction site, and they are palindromic. Restriction enzymes are used to know the order of the gene in the chromosome. Second, what is the chemical structure of the gene, and what is the sequence of the DNA which regulates the function of the gene. And third, to create new genetic combination. Nomenclature. Here I am giving one example of ECO R1. So this ECO R1, so it is obtained from the E. coli, which is present in the colon of the human beings. So E stands for the genus of the organism. CO stands for, that is, CO means two, it is written with the small letters and first two letters of the species, that is coli. And R stands for the strain, that is the rough strain, and for denoting it, capital letter is used. So R stands for rough, And the last, that is it is Roman numeral 1, and 1 stands for the order in which enzyme is isolated. How does restriction enzyme work? First, it inspects the length of the DNA molecule. It looks for the specific sequence present in the DNA. It finds the specific sequence. After finding, it binds with that particular site and cut the DNA in their sugar phosphate backbone. This is how restriction enzyme work. We will see here with the example of EcoR1. Certain restriction enzymes, they cut DNA molecules little away from the palindromic site. For example, EcoR1. This is the sequence where EcoR1 acts. It cuts the sequence, this palindromic sequence, between G and A. It is cutting the DNA in such a way that it comes to possess sticky end. So same enzyme works on the foreign DNA and it comes to possess this sticky end. Why this is called sticky end? Because it forms hydrogen bond with the complementary cut counterparts. That's why it is called sticky end. Here, these sticky ends, they initiate the action of DNA ligase. As a result of this, recombinant DNA is formed. So this was about the restriction enzyme which acts little away from the palindromic site. Other restriction enzyme, that is SMA1, it is obtained from Serratia marcescens. And this restriction enzyme, it acts in the middle of palindromic site of DNA duplex. It splits the DNA molecule in such a way that ends which are formed, they are called blunt end or flush end. When DNA of a vector and the foreign DNA are cut with the same restriction enzyme, they come to possess same sticky end and they are joined by DNA ligase. And the recombinant DNA can't be formed unless the vector DNA or the foreign DNA are cut with same restriction enzyme. When they have same sticky end, they are ligated or joined by DNA ligase. So here the next enzyme, that is DNA ligase, is used to link the DNA fragments, and the most common ligase enzyme is T4 DNA ligase. The other enzyme is DNA polymerase. DNA polymerase helps in the synthesis of new DNA strand in the direction of 5' to 3'. So this was all about the enzymes involved in the recombinant DNA technology.